What I need to do is calculate out the A and little a, right?
Yeah.
So this is 0.25
Oh, big
plus
and little a. Okay.
1/2 of 0.5, which is equal to 0.25, 0.5, right? This one, 0.25 plus 1/2 of 0.5 equals 0.5, because we take half of those. So then I calculate this out, I do 0.5
Squared
squared.
plus 2 times 0.5 times 0.5 plus 0.25 squared or 0.5 squared. Okay.
Okay. This was not punishment for your exam. This is punishment for Wednesday coming up. What's this Wednesday? Anybody look forward? Look ahead. Meiosis comes back.
Ooh, yeah.
We're doing
A?
meiosis this Wednesday.
I
So
knew
we need to
that.
know, remember, meiosis, mitotic division, and Hardy-Weinberg. Okay, so this is a reminder to do some refreshing. Because we really need to do some refreshing after the cell division question, right? We got this.
Oh,
I was
yeah.
actually really impressed that the only 2 people. I threw that on there thinking that was super easy. I needed 5 additional points or whatever. And then 2 whole people got it right. I almost thought about just giving them an A in the class.
Yeah, that's what I was gonna say.
Like boom, they just get it.
Whatever.
With that said, this week's lab we're going to do respiration. We're also going to do the antibacterial lab. So we as added. You don't need to do a prep for that. Any questions? Okay. I would like you to now draw. Okay. Oh, next question. What are tRNAs and where are they synthesized? Doctor, what synthesizes tRNAs? What are they? What are they made? Where do we do this at?
tRNAs are made from
I think they're made by ribosomes. Either that or the
DNA.
or the one endoplasmic or one of the endoplasmic reticula.
I have that down for the exam.
Were you right on?
Pretty sure.
Wasn't that the right answer? Because I think I just.
Aren't they made in the RER?
Ruffy
Yeah,
are.
2 or 3. Like it was really sad.
With the ribosome, like you said?
What are
Add-
tRNAs?
Well, amino acids attached to the. I mean,
They're a
And-
RNA that has an amino acid attached to it.
Yeah.
We find them where? In the cytoplasm.
A
Excellent.
cytoplasm.
Uh-huh.
Okay.
We find charged ones in what organelle if you're a eukaryotic organism? Rough ER. Excellent.
oh, there you go.
Because what's being made in the rough ER? Proteins. Excellent. We're making polypeptides. We're making proteins. So we're going to assemble them with what organelle or enzyme? Ribosomes. So we assemble things, the polypeptides with the ribosome. We're going to make tRNAs with genes that make mRNAs. That'll be the RNA, and then we can attach an amino acid to it based on the R group attachments. So there's genes that regulate those. Those are going to be constitutively made because we're going to make them all the time.
All the time.
Excellent. Things that are regulated get positive and negative regulation. It means we can shut it off or we can turn it on, make it go faster. We make something go all the time. Constitutively, we're going to make it fast or slow depending on the promoter. What makes a promoter go faster? Dr. Cho, what makes a promoter go faster?
Catalyst.
What makes a promoter go faster?
Not a catalyst. How do you take this out of here? Do you know?
So what makes a promoter go faster?
I haven't done it last.
Oh, thank you.
It's strong.
Strong promoter. Okay.
What makes it go slower?
Weak promoter.
It's weak. What does it mean to be a weak promoter?
Lots of G's and
You said
It is
C.
not G's and C's.
Oh,
G's and C's could be it, but it is not. All it is a change in base pairs. It's a specific sequence. So a strong promoter is a specific sequence. It could be that your tata would be a strong sequence. So in that case, a GACA would be a weaker promoter. But it doesn't have to be that way. A strong promoter is just a very specific type of sequences. So why would we regulate something? What's the purpose of regulation? Not waste energy, right?
Yep.
That's the
Not waste
whole
energy.
reason to do this. And where do cells get their energy?
Carbohydrates?
Cellular respiration.
Oh.
Excellent. What are we doing this today? Cellular respiration. Okay. So, talk to each other. What is the ETC, the electron transport chain, and draw it out. What complexes do we have? What did we do last month?
What did we do last week?
No, I don't remember what I did last Wednesday, so that's a hard question. But what did we do last Wednesday?
I don't remember.
Think about the same.
I don't think I would understand any of the things. Just like that, I need like
I love the worksheets too. Yeah. Actually, good practice.
We're going to create membranes. So this is my inner membrane, that's an outer membrane. I am going to have pumps. Complex 1, 2, then we have this Q thing, right, that zips around. Complex 3, and then we have site C, and complex 4. Now that we have it drawn out, go through what are our products, reactants, and what is happening.
Are you getting that from the lecture?
What's the goal of the electron transport chain? To create a gradient. Nice job.
To create.
That's an excellent answer. To create a gradient. I need something in here. What am I going to put in here?
NADH?
Uh-uh.
Oh no.
Hydrogen, I need to put protons.
Protons.
Lots and lots and lots and lots of protons need to go in here, okay? I mean, an absurd amount. How do I get protons there?
Water?
I gotta put them there, right? I gotta push them there. This is what type of transport? Active.
Active transport.
We're gonna use energy to do that. So I'm gonna drop something off and pump a proton across. What am I dropping off? Excellent. NADH
NADH.
gets dropped off. NADH. Jesus. gets dropped off. That's going to make NAD+. The electrons are going to get dropped off. Those electrons are going to go through from high to low energy. Okay? Then we're going to drop something off at complex 2. What do we drop off at complex 2?
FADH2.
FADH2. To make FAD. I think it's FA.
FA
My spelling is just
DH2.
horrible. I was telling Jeff this morning, I was like, such a beautiful, gorgeous morning. He's like, why did you show up? I was like, that's a good question. I did something wrong today. Came here. Anyways, and I came here to that. But, we drop off that pumps more electrons, they re-energize, and we pump more protons across. Those electrons are received by what molecule? Talk to Sheriff. Those electrons are received by what molecule at the end?
Yeah, that was what I was thinking.
What receives the electrons at the end?
O2.
O2. So we take that O2, combine it with what?
Two protons.
Yeah, some protons to make
Water.
water. That's all we've done. That is the electron transport chain. We created our gradient. That was the whole purpose. I made a gradient. There's a high amount of potential energy now in here. This is called electrochemical gradient. Because I have a bunch of protons on this side, and what I got on this side?
Electron.
No protons. A lot of protons, no protons. Okay? Do you see anything to allow protons to come back across this way?
No.
That is not a part of the electron transport chain. So if we were to continue on looking at this membrane, go further down the membrane, there's another little complex. What is that called?
ATP synthase.
Excellent. ATP synthase. What does ATP synthase do? Synthesizes ATP. Okay, so we take ADP and
Inorganic phosphate.
ADP and add
plus inorganic phosphate and make
ATP.
ATP. And so what we're doing is all of those protons that are there are going to power that. And when it goes through, it's going to literally turn. That enzyme, because its active site is going to be a certain shape, is going to grab that ADP molecule. And because inorganic phosphate has a charge, it's going to grab a hold of that as well. And when it turns, it's going to smash them together. And that's going to make ATP. That is the purpose of this. So, what is the purpose of the electron transport chain? To make ATP from an gradient. Excellent. That's it. So I have a gradient. I make ATP. Does this have anything to do with, at this point in time, with glucose, glycolysis, anything like that? That's independent. Okay? So glycolysis and Krebs and all of that is happening independently of the electron transport chain. The electron transport chain is where you get your ATP. So what happens if I were to disrupt the electron transport chain? You die. Really quick.
The electron transport
Literally
chain adds 2 seconds.
About
to
2
it.
seconds. That gradient collapses in about 2 seconds. That's impressive. Okay, that's a very impressive amount of efficiency. So how am I going to recharge it? Well, I'm dropping off NADHs and FADHs. Where am I getting those? That's where glycolysis and the Krebs cycle comes to play. So you ate breakfast this morning. Okay, that has a lot of potential energy in those chemical bonds. I got to break those chemical bonds so I can build an electrochemical gradient using my NADs and FADs. That's what we're going to make. So today we're going to try to understand glycolysis. Glycolysis is the process of taking glucose and turning it into pyruvate, NADHs, and FADHs. You're also going to gain ATP out of this. This is called substrate level phosphorylation. Substrate level phosphorylation, just by breaking glucose apart, I can steal energy from it. Okay, I'm going from lower energy state to a higher energy state. I'm just going to steal some shit from it. And I'm going to steal an ATP molecule. But to do that, I got to put in energy as well, because it's glucose stable. Yeah. If I have a pile of glucose right here on the table, we leave, we come back a year from now, what's going to be sitting here on the table? Glucose. Perfectly stable. So what I got to do is I got to add something to the system. I got to charge it. Okay? We're going to add ATP in. And then we're going to break those bonds apart, and we're going to steal more energy than we added into it. That is glycolysis. All right? So my meme this morning was actually very relevant. Anaerobic respiration has only to do with glycolysis. Okay? I'm still going to gain ATP. I'm going to gain energy just by breaking down glucose. Just the fact of breaking it down without using the electron transport chain, I'm going to gain energy. But if I now substitute in the electron transport chain, I send those FADHs and NADHs off, I steal a ton more. So one glucose molecule produces about 30 to 36 ATP. It's pretty good. So that's what we're going to understand today. How does glycolysis work? And then we're going to start going into Krebs. Now, what do I want your comfort level this weekend? Okay? That's like the real question. Today, after class, you should have a reasonable understanding of what happens. At this point, you should know what happens in the electron transport chain. We should have a reasonable understanding of what happens in glycolysis and Krebs cycle. After lab, you either know it or you don't move on. That is where we're at, okay? So you stay there until you know it. You're going to know every single mechanism, so forth. So that's our comfort level. So, okay? So that is where we want to be at this week with it.
You said electron transport chain.
You should know that really.
We should know
comfortably
that,
now.
and then
You should be okay understanding the reactants, products, and how things work with glycolysis and Krebs by the end of today. And then on lab, you nail that shit. Every single enzyme, you know where it's going, how it goes in and out, and so forth. And don't worry, you will get there. You never had to not get there, okay? Just like meiosis, which came back and we all forgot. It is.
Yeah.
So you will know meiosis again because what's happening this Wednesday? Meiosis. Because what is our purpose starting next week?
to create the whole organism.
We're going to make the organism. We're going from right now, we've assembled DNA. We figured out how to assemble DNA. Now we got to figure out how to power the system. Once we have those two things figured out, now we start building. And we're going to start doing some really cool stuff as we start building up. Okay? All right. Let's get started with worksheet.
Ooh, yeah.
Make sure to work together.
Okay.
Don't leave people out. Make sure to work together.
Oh, yes.
Refer to Model 1. What molecule from food is primary reactant for glycolysis?
Okay, so potential energy and then glucose, pyruvate.
How many carbons are in the reactant molecule?
So what molecule from food is the primary reactant for glycosis? Glucose?
Oh, glucose. Okay.
How many carbon atoms are in the reactant molecule of glucose? That's 6, right?
Yeah, C6H12O6.
Yeah.
Carbon atoms from glucose end up in pyruvate as a product of glycolysis. How many carbon atoms are in pyruvate? 1, 2, 3. How many pyruvate molecules are made from each glucose? C3, H3O3.
How many pyruvate molecules from each
Then
glucose?
we're gonna go in
So just one molecule? Or, huh?
Stovzem.
Because this?
Yeah.
Okay.
Yeah, it looks like there are two
So two.
pyruvate. Yeah,
Okay.
two pyruvate.
Times 2. Okay. Does the process of glycosis require an input of energy?
Provide specific evidence for Model 1.
Well, that's a yes, right? And the
Yeah.
evidence is that's the process of glycosis. From glucose to phospho.
It needs to steal energy from glucose to make ATP. It needs energy.
Is what he was saying here. Glucose is stable and it adds ATP to break apart and steal.
now I don't have to fill this out. But you need to figure out your
Yeah. So like you need the, it's asking.
Yeah, but it needs first.
Yeah,
We're gonna go
apart.
Well, I think it's, oh, is the energy consistency?
So what does it break apart?
just taking a guess. I don't know.
Yeah.
Because you're only sitting at 6190 credits.
What's that 1,3 biphos? What is that doing? This box over here? Nothing?
You need one more social.
I think that's just when you get the NADH out.
Is that one, is that for food?
NADH. Propulsion expenditure by the author of this activity could be the highest one. Would you think that he put it up there because that's like that curve, you know, where it goes like this?
Yeah, sort of.
Is the energy level? Yeah. Does pyruvate have more or less potential energy than glucose?
Refer to Model 1. Propose an explanation for why the author of this activity put P-gal at the highest point in the Model 1 diagram.
I thought highest energy level, like in the diagram. You know, like
Oh yeah. Yeah, sort of like the
right.
Yeah.
And then 5. Does pyruvate have more or less potential energy than glucose? They have the same, don't they? Would you say the same?
Mm-hmm.
Same energy? Glucose and pyruvate? Oh, if they're in half, maybe, no. Would you say the same? They have the number 5.
I was thinking they'd have less.
Pyruvate is less energy than glucose.
Less because it's gonna be
2 divided by 2.
no, also because they have less like non-polar bonds.
Oh. Is that why you think it's less also? Because of the non-polar bonds?
Yeah, there's just less bonds in general.
Less
Oh,
what?
yeah.
I think less bonds in
Less bonds, okay.
Yeah.
Below the starting.
Less bonds and non. What is the net production of ATP by glycosis? I don't understand that. What did you put? Oh, you're still on that?
Less bonds overall.
What is the net production of ATP by glycosis?
What is the net production? Glycolysis. I think it like produces 2 ATP, like that meme he showed us.
Oh, because it's like here is 2, and then here is 4, or what?
Oh, okay. So that's probably like 4 ATP. Then
But it had 2, so then it, I don't know. What
Well,
do you
it
think?
looks like it got 2 and it came out 4.
So then 2? If you already had 2?
I
Don't know. What? Let's go to the next. What molecule acts as an electron acceptor in glycosyl?
I think it's the NADH.
NAD plus electron acceptors are bases, which take protons. So,
In the last steps of glycosis, 4 ATP molecules are produced. Analyze model 1 to find the source of the 4 inorganic.
4 inorganic phosphates that are added to ADP to make 4 ATP molecules. Describe the origins of the 4 inorganic phosphates here. So there are 4 PO4 free minuses.
There's, I see one. Difficult.
2 PIs, 2 organic phosphates,
Oh,
with the
times 2.
NADH. Yeah, and then, and I think when
Oh, in the last steps. Okay. 2. I don't know, could it be these 2 and then these 2? Is that 4?
1 free BPG, 2 NAD plus 2H plus 2 PI,
Oh, the origins of.
No, it's 2 ATP because it uses 2 ATP. Oh, it's 2. It's a net so it says on the computer.
I know, where else would everybody?
I don't know. Do you? How about that one? Do you know number 8?
how are you able to answer number 8?
I'm thinking, but I'm not sure.
I'm thinking it was like NADH and 2 NADH and 2 ATP.
I said it came from the BPG.
Oh, yeah.
DPG?
And I also put the BPG in consideration too.
Okay, I don't see a DPG.
It's the one on the top right.
Yeah, it's that one on the top right. Yep, there you are.
This one here?
Yeah, that's the
So
one
this
for
is 2 right here, right? Times 2?
Yeah, there's 2 of those.
Yeah.
So there's a total of 4 phosphate groups.
Yeah.
Because there's 2 times 2, or no? Yes, okay.
And where did the other 2 come from?
Well, it's 2 times 2.
Oh, okay.
That's what I understood. But I don't see times 2 there. That's what's.
Yeah, it is times 2. 1 free BPG times X2.
Oh, okay, I see it now.
Yeah, so the four of these came out of the 1 free BPGs. Yeah.
Okay, model 2. Outer membrane.
I got 4 and I should have I said it was really
How is this one? Mitochondrium?
According to Model 2, where in a cell does the link reaction take place?
Like my ass.
Link reaction. Pyruvate and acetyl. How do you say acetyl? Acetyl. Acetyl. So would you say between the pyruvate and the acetyl is the number 9? Where in the cell does the link reaction take place? Inner matrix?
Yeah.
Oh, well, that makes sense.
In the inner matrix.
Obviously, my boyfriend and but like a lot of college students can't live on campus.
Consider the pyruvate molecule. Is the pyruvate molecule?
Yeah. Work in different I thought you meant like it feels very like it feels like very
What happened? Why did you laugh?
Oh, math. They were talking about math. The math classes.
Oh.
The lowest one was 142.
The lowest math class offered at the university are
Yeah, the lowest
Oh.
math class offered at the university.
142.
I took 141
We have a
at
question.
the college.
over here trying to convince you to do something. I know, I didn't.
So
I
we
have a question for you
Oh,
guys.
there's a question. Okay.
Actually, how soon is that?
I'm
Is
listening.
carbon oxidized in this stuff?
No. Sorry. Well, don't worry. I could be wrong, right? Oh, wait. No. Nope. Reduction. Wasn't there French online? French online? For well, Italian is kind of like Spanish.
There you go.
So I can kind of help you there.
You can kind of.
Registered classes today?
Yes.
We did
Alright.
register for classes last week.
10 seconds.
Oh, I'm like.
I guess I'm not listening.
Going. My OChem people, yes or no?
Yes.
Yes, thank you.
Yes. It's
Isn't it going
We're
down?
adding a hydrogen. The oxygen is losing electrons. Added a hydrogen.
Oh yeah.
Thank
There we
you.
go. There we
We
go.
added a nitrogen? Where did we add the hydrogen?
Mar, look at the diagram real closely. There's a C double bond O.
Yeah.
But then in the second diagram, it has a CHOH instead of C double O.
But it's going from down up. It's taking it away.
NAD,
It's losing electrons.
NADHs.
Explain later.
Okay? Go through. Tell me, what are the different components of this molecule? NADs, NADHs. What are the components of this molecule?
Components, phosphate groups. Ribose.
What kind of bonds do I have here?
Phosphodiester bond.
Excellent. We have sugars, right? RNA or DNA?
RNA.
RNA, because we have an OH and carbon.
2.
There we go. What is this?
A nitrogenous base.
Excellent. Pyridine or pyrimidine?
Purine.
There we go. Oh, things you guys learned. Wow. Holy cow. And then we have another nucleic acid here. And here we have an H. All right. So, what is the purpose of this molecule?
Transport protons.
Excellent. Transport protons and electrons, right? So when we have the proton, we also have the electrons present as well. Okay? Keep going.
Consider the pyruvate molecule. Is the pyruvate molecule likely to move across the mitochondrial membrane by
You actually
diffusion?
don't have to write an essay if you have all this.
Yeah. Really? No, what like summers are like a little long
I wrote my essay just in case, and my GPA is 3.5.
I wrote one.
Yeah.
I think I go in because
Well, okay. That's not a question, is it? What are we doing? Oh, it's just over here.
and then they'll provide because there's so many apps.
Where are you on 10? Consider, or what are we doing? 10, right?
Number 10.
Oh, you're way past 10? Okay. Is the pyruvate molecule likely to move across the mitochondrial membrane by diffusion? I don't know what's diffusion. Diffusion?
Like facilitated diffusion. There's like simple, facilitated, and active transport.
Which should include polarity. Oh, okay. So it goes from the pyruvate to the acetyl.
And like sometimes like they have like additional questions in college. But I think I just
Well, it lost polarity, doesn't it?
Well, it looks like pyruvate is an ionic molecule.
Yeah.
With the formula C3H3O3 minus.
you can find it on
Ionic.
the website, but you're
Well, I don't know. I just know that the pyruvate is polar. Ionic polar. The pyruvate
Number
is ionic.
10A.
Oh, number 10, yeah. The pyruvate is ionic polar, right? And the
Yeah, pyruvate-
acetyl. Say it again. Acetyl.
Acetyl.
It's it looks covalent, right? So it looks like it changed or lost polarity. What would you say? But diffusion, I don't understand the word.
Diffusion is passing through the membrane.
Yeah.
So yeah,
I'm starting with what molecule?
Glucose.
Glucose. I start with glucose. How many carbons do I have?
6.
6 carbons. I'm at a relatively decent amount of potential energy. Okay? Nothing great, nothing, fabulous, but it's also very stable. I'm going to put in what into the system?
ATP.
2 ATP. I'm going to put in 2 ATP, and that's going to do what to the molecule?
Add a phosphate.
It's going to add a phosphate and cause it to break apart. We're going to cleave it in half. And so adding that phosphate is called pGal. And pGal adds energy to the system. It makes it a higher energy state. So I've added energy to the state and I broke it in half. And when we do that, now all of a sudden I can do substrate level phosphorylation and steal ATP from that molecule. I'm going to get an NADH per pGal and 2 ATP per pGal. And you have 2 pGals because I broke glucose in half. I end up with what molecule at the end?
Pyruvate.
How many carbons?
3.
3 carbons. So I have 2 of them. So 2 times 3 equals
6.
Have I created any CO2? No. Have I actually even lowered the amount of energy in the system? No.
No.
It's the same. Now this is cool. I put in glucose. I'll get you in a second. I put in glucose and I steal shit out of it. I stole 2 ATP and NADHs. And I steal 4 NADHs. So I get my net of breaking glucose apart is 2 ATP and 4 NADHs. That's just from doing it. So I'm going to steal those 2 NADHs there, and I'm going to steal 4 ATP there. Why do I say I have a net of 2? Because I put 2 in.
You use 2 ATP.
So I put 2 in, 1 sec, and I get 4 out. Okay? So the return on investment is fabulous. Because did I change my amount of energy available? All I did was break it apart. I took a molecule, a big molecule, and turned it into 2 small ones, and was able to steal energy from it. This is the purpose of glycolysis. Now I have something I can actually use. It's called pyruvate. How big is glucose? That's a big molecule. That's a big molecule. Should glucose pass through your membranes? No. If glucose passes through your membranes, what do you have? Excellent.
A what?
You have diabetes.
Diabetes.
It's all good. We're America, it don't matter. Okay? So that's not good. That's passing through your membrane. So I don't want it to be able to go through, but pyruvate now is smaller. It's smaller, but still charged. So it doesn't really want to go through membranes, but I can drag it through a protein channel. I can do something to it so that it'll go through. What I'm going to do is I'm going to attach an enzyme to it that's going to bring it through. That's called your linkage step. All right? So, purpose of glycolysis was what?
Break glucose
Break
into 2
glucose down.
pyruvates.
All we did. Break glucose down. That was it. In the process, what did I gain?
ATP and
4 NADHs and 2
ATP.
ATP. What did my meme say? 2 ATP. This is cool. If you are a bacterial cell, does this work for you? Yes.
Yeah.
Fine. Bacterial cells do what type of respiration? Anaerobic. Excellent. That's all they do. We're doing anaerobic respiration. We're going to make a little bit. We're going to gain it. But does that work for you?
No.
Because how much more activity are you doing?
A lot more.
So I need to do something else. So I'm going to use what's left over, that pyruvate, to actually gain something. Now, let's imagine you're a plant, and I have an excess amount of pyruvate. What can I do with pyruvate? Turn it into
Glucose.
glucose. What do plants make?
glucose.
Glucose. That's it. So those reactions are equal. Can this reaction go in either direction?
Yeah.
Absolutely. Just run the enzymes backward. Okay, so I'm gonna work my way back. So
Question 5 asks is if there's
the
more or
inspector
less
can give you an actual question.
You don't know?
if there's more or less energy in fluid than glucose,
Which one?
if it's number 5.
Yeah, number 5. So it's the same.
But that's if there's 2.
That's correct.
Oh.
It's correct. But because we have 2, it's the same net. So, what was your question?
Is BPG less potential energy than PGAL?
It's counter bonds.
What question is that?
So
Okay.
this is actually a good question because it brings up a very valid point in biochemistry. In other forms of chemistry, you have to figure out what each bond represents. Don't care about that shit. Carbons, hydrogens, is all we care about. Carbons and hydrogens. So let's count our carbons. 1, 2, 3. 1, 2, 3. Okay? How many hydrogens do we have? 1, 2, 3, 4. 1, 2, 3, 4. So it is
Same.
same. Don't question biochemistry.
What about the bipolar
I lied. 1. Don't tell Samina. 2, 3, 4, 5. 1, 2, 3, 4. That one has more. Damn it. Try it.
Count your H's.
Count your H's.
So P-gal
I'll
has
say it. That's the difference.
count your fucking H's.
Because it count your fucking hydrogen doesn't work. Okay, keep going.
Before that, you give us
Yeah.
You could be like, oh, you guys all do one of
Never get 50 on Oh wait, I got 51.
Correct, you're adding
Okay, that was like a one-time thing though, and I was cooking, right? It was like,
But I actually knew what I was talking about.
yeah, I lost. Last time I said something
What?
weeks ago. It was the
You're gonna take those ATP back, but when you put that ATP in, you literally break it in half.
So for 10, what method would you propose? Which a pyruviate molecule moves across the mitochondrial membrane. Oh, that's fine.
10B
Yeah.
or 10A.
Well, 10A, pyruviate is ionic polar.
Ionic.
Well, is it likely to move across the mitochondrial membrane by diffusion?
Yeah, because it's small.
So it looks like we're on linkage. Okay, let's take one more minute to debrief. I thought we were still people were catching up. Okay, so this is actually all the steps of glycolysis. So what happens at every one of these arrows? A reaction, right? So what is catalyzing every one of these arrows? An enzyme. Okay? So if I have this, can I make this? Yes,
Yeah.
absolutely. If I have this, I can make
That.
this. That's just an enzyme reaction, so it's not going to take any energy to do that. But I am going to take energy to break apart this very stable molecule. So the first thing I'm going to put in is ATP. And then I'm going to do a rearrangement. And then I'm going to put in a ATP.
Another ATP.
1, 2. How many net do I put in?
2 ATP.
How many do I get back out?
4 ATP.
Okay, there we go. All right. So then I'm going to get this to be a high energy molecule. This is what molecule?
BPG.
Yep. Then I'm going to, so let's just start this. This is glucose. And this is pyruvate. I'm going to here add another phosphate, but this is where biology can get a little funky. I don't need to put an ATP molecule on it. I can actually just put an inorganic phosphate. And that's going to allow us to steal
Is
some
that good?
stuff.
Yeah.
You good, Carter?
Sorry.
Yeah, you're good.
I guess I'll put it.
Sorry.
Yeah, that happens. Hey, at least we're fairly handicap accessible around here. There's a lot of campuses that aren't. It's pretty awful. So then I'm going to start stealing things. And I have some extra protons here that we're going to steal. So if I'm doing that, I'm going to gain in NADH.
Mm-hmm.
And I'm going to do that here as well. I'm going to gain in NADH. So I put in 2 ATP, I get out 2 ATP, and then I get out, what did I lose right there?
Hydroxide.
An hydroxide was there. That's not what I lost. What did I lose right there?
Phos.
From there to there, I lost.
A phosphate.
So I'm going to go from ADP to ATP. I'm going to gain an ATP. From right here to right here, what did I lose?
A phosphate.
So I'm going to gain in
ATP.
ATP. So I get 2 ATP back per one of those. So what's my net total ATP getting back?
2.
2, because I get back 4, but I put in 2 into the system. So how much does glycolysis give you for ATP?
2.
2. Just off that alone. Okay?
Where's the phosphate?
Just there. Just there.
It attaches?
It's just there. It's in the environment. Inorganic phosphates are actually everywhere. It's just salt. Yeah. Okay? Question for you guys.
Step 5.
Okay, 10 seconds. All right, this is a hard question, so time for sure.
I thought that molecule has too little energy.
I did too. But which is, first of all, step 5? How do you count the steps?
Step 5 is the last step.
That's what I thought. But
Oh yeah. That's step 5.
So
All
then
of that
4
down there. And step 4 is the glucose with your inorganic phosphates on it.
So this step 5 comes out of that one, right? Just
Yeah.
this side, only from this side. And it needs more phosphate, right? I guess. So that's why I thought it had little energy. But I don't know.
The next
could
step,
have
does
too
it
much.
matter about the energy? No, it has nothing to do about energy. This has to do about
The change.
shape.
No.
Because I need to be able to recognize
I
it for
should've
the
saw
next
that.
enzyme. Okay, so enzymes are going to regulate this. Enzymes are going to perform all of these things. And so if I have a high amount of ATP already, do I need to make more of it? No. So ATP will slow down the reaction. If I have low amount of ATP, what's going to happen?
You want
I
more
want
of
to
it.
speed it up. So if I have less ATP present, I'm going to make the binding site have a higher affinity. We're going to make it run faster. So this does not have anything to do about energy of the actual molecule. It has to do with being in the right shape. Because what do I ultimately want? What molecule?
Pyruvate.
So,
Okay.
are you saying the ribose, or sorry,
It's actually like a fructose, I think.
yeah,
Yeah.
that, is that the correct shape that it wants to be in?
Yeah, so it doesn't matter.
Okay.
These shapes, this is not the correct shape. I need to be able to break this apart to make it to the right shape. The right shape is pyruvate.
Oh, now
Okay?
it makes
But it needs to be in the correct conformation so I can do this. All right, so this brings up another good question. You did not eat any glucose this morning for breakfast. I don't know. I'm just taking a guess. But you ate fructose. Make any guess. What happens?
Your body uses the other carbohydrates.
No, you use fructose. I jump in here.
You run
I don't
on.
have to start at this spot. I can put the molecule in here. I can put it in at different spots. So let's say you decide you want to do the keto diet. Okay? So we're going to do the keto diet. The keto diet is we're putting in ketones instead of glucose. So are you going to be able to go through the glycolytic process? Oh shit. Now this has a problem. Does it matter if your body needs to go through glycolysis first?
I guess it does.
Why? Do all cells have a mitochondria? No, not all cells have mitochondria. Does anybody know what cells do not have mitochondria? It was in the reading.
Prokaryote.
No.
Oh no, prokaryote.
In your cells. Talk to you. Try to figure this out. Which cells do not have mitochondria?
Which cell? In our cells?
Yeah, in our cells. I read it and I don't. I did this reading on Wednesday, so I don't remember. Yeah, reading. I did this on. I did all the reading on Wednesday.
Anybody remember now from the reading? Which cells do not have mitochondria? Red blood cells.
Oh,
Red blood cells do not have mitochondria. So what's the only way a red blood cell can make energy? Through anaerobic respiration, through glycolysis. So if you do not have any glucose in your blood because you're doing the ketone diet, can your red blood cells make any energy?
No.
No. So what happens to red blood cells?
They don't.
They die. Does anybody know what happens to people who are on the keto diet?
anemia.
They feel tired and sluggish and weak. That's because they have anemia. Oh shit. Okay. Now we're starting to get somewhere. So, for those people, kinesiology and dietitians and physical therapists, you want to be able to start understanding how to actually do dieting and proper nutrition and all those. We need to understand the cellular level. We need to understand the mechanism. So being part of this and understanding glycolysis is really important. Okay? So,
If you do fructose, that means eating fruit and
No.
No, what is it?
no. Fructose is the shape of a sugar. It's just a different type of sugar.
Okay.
It's like maltose, lactose.
So
So let's say you drank your milk this morning. What was the first step?
break the
Excellent. What enzyme in your body broke down
lactase.
Lactase, beta-galactosidase broke down the lactose and made it what?
Glucose.
Galactose and glucose. And then we take
Glucose
the
plus
galactose and turn it into glucose. So we would add, if we started with lactose, we'd have added another enzyme, another step. All we have to do in this pathway is take steps away or put new steps in, depending on where we put it. Let's say you ate maltose this morning. Okay? You've added another step up here. So you'd have had it maltase, would have broken things down. And then you'd have had a blah, blah, blah.
You would have had a lot more.
Right? You had a, what?
So what do you eat to get more?
You've been in fruits and sugars.
A beer?
A beer.
Maybe beer, I don't know.
Anybody got a
All
beer
right,
for
that sounds
breakfast?
great now, let's do this.
Let's try it. Can we do that in the lab?
Beer.
I don't
No.
even drink.
We're gonna learn not to do things like keto diet.
Yeah, no keto
So,
diet.
but the question, fructose is okay to do.
Anything is okay to do.
Except-
That's the point. If you have the enzyme to break it down, you can metabolize it. Why is lactose, if you're lactose intolerant, a problem?
You can't break
You
it.
don't have the enzyme to break it down. People that are allergic to alcohol, why is it a problem?
You don't have the enzyme.
You don't have the enzyme to break it down.
Oh,
But as long as you have the enzyme, who cares? Use it. So then my next question, if your body is low on glucose, what does your body do? It makes it. All reactions are reversible. Therefore, I can make it. Let's imagine your body is low on vitamin, I don't know, I don't care, K. B12, B12, low on B12. What does it do?
It makes
It
vitamin.
makes it with enzymes. So, do you need to be supplementing these things? No. Okay? So,
But then
we're going to
there-
get to that. We're going to get to the metabolic pathways. Yeah.
Is it beneficial or like?
No, because you already had the
you're doing
enzymes,
your job.
right? And if you didn't have
You're,
lactose
you're
present,
good.
would your body have made the enzyme?
What was the question?
No, is it cost or benefit, right? So why did we do gene regulation? To be able to regulate this. Why make the lactase enzyme if you don't have lactose in your diet? Why do those things? Well, if I need it, because if I need glucose, I need glucose, right?
Yeah.
So I'm going to build that extra enzymes that are up there. We're going to regulate them. We're going to regulate them with the gene regulation. Make the enzyme or not. Then we're going to throttle the enzyme to make it or not.
Which site? I'm a regular and I must have a higher affinity for ATP.
I want to say. I'm gonna go with it.
Okay? Talk to sure.
I'll put the two first ones there.
I put C and D. I put both sides have equal affinity and or neither. Equal affinity.
Could be. I was between all of them, but I
Which
just.
site is absolutely needed to be able to bind?
Active.
The active site. Therefore, the active site would be a higher regulatory function.
Oh, there we
We need
go.
the regulatory to be able to leave and come as it goes.
So if our body makes it, why do we supplement it?
That's a fabulous question. We're going to answer that here soon.
Okay.
Do you? So
Good
I think we
question,
talked about
Barry.
this in last quarter. What happens in your life if your body makes things? Why do you supplement it? Did we talk about vitamins last quarter?
Yes.
What happens to your life expectancy? Because she wasn't in the class. What happens to your life expectancy if you take a vitamin? Goes up or down? Goes down statistically.
What?
Vitamins are terrible for you. Absolutely horrible for you. So vitamins are not regulated by the FDA. There is no regulatory component to them, and we don't actually know what's in them. But we do know where they're manufactured, and they're not usually manufactured in the United States, and therefore they have things in them like heavy metals, so forth. So the average person should not be taking a vitamin. Most vitamins actually decrease your life expectancy and increase your need for other things. But it also might give you a false sense of security too. A well-balanced diet is much better for you.
Placebo.
I feel like my
placebo-
zinc works pretty well, but now that's disappointing. Okay.
Just go for it. Let's talk about ethics for a second, okay? So, because I think we talked a little about this before.
Yeah, you did.
But a mom thinks that taking vitamin C is going to prevent her from getting sick. Vitamin C has nothing to do with getting you sick or preventing you from getting sick. So mom, poor, needs to not get sick, goes out and buys vitamin C, spends her money on vitamin C instead of buying good food or being able to do other things. Is that ethical of the company to do it? No. So most of these things are completely unethical as well.
Could you remind me again why organic is also racial expectations?
Yeah, organic food also decreases life expectancy for very similar reasons. When they say they do not have a pesticide on them, that is full of shit. They use things like arsenic and so forth.
And literally S.
Yes, literally.
They use S.
Yeah, they
Or
literally use shit on it.
I was talking to someone and they were trying to say that they were trying to tell me that consuming the pesticides versus consuming the heavy metals, I guess that consuming the pesticides is like a higher cost. It's like more deep. I don't know what they were trying to say. I didn't really listen.
Probably made no sense. Let's just go with it. Made no sense.
Have you
So at
watched?
some point, you should just start figuring out all the chemistry and just being like, that chemistry doesn't make sense.
Have you
That's
watched
where we're at.
Rotten? In Netflix, Rotten. It's about all the industry.
So,
I think I've watched that.
it should be on the link.
I work in pharmacy, so I've watched a lot of the pharmaceutical shows.
I'm on the link.
Rotten
Link
is-
reaction.
is more about the
10B.
But yeah.
There's
it's really
one
good.
that you should watch. I'll find it and
I'll
I'll
send
tell
it,
you at
okay?
class. It was really good.
Grace, so on Canvas, I think it's in this section too, I don't know. There's a thing called neat links and articles and a whole bunch of citations. Some of those citations have that.
Okay, I should go look at it. My family's a really big vitamin family. Like,
They're very much
Yeah,
wasting their money.
my family too.
but I feel like zinc works.
One thing that I've noticed, like for nails, I noticed that when I take biotin and that it works, but it's not great, but but I can see it might work like
So
a little
you're
bit,
a scientist.
maybe a little
And so
bit.
maybe a tiny bit is working.
Yeah,
And so let's say it's helping with one thing, but hurting other things.
yeah,
Yes.
Yeah.
And then you're also a scientist, you know things that are true about the placebo effect.
yeah.
Yeah.
So do they just like make them so that it degrades in the other parts of the food, or do you spend more money on more vitamins that you don't need?
No, just
Make money.
Just garbage.
Just make money.
There's, if you have a reasonable diet, there is no reason to take any vitamins or supplements.
But do the vitamins, do some vitamins like
Arsenic in my diet.
Would you want to
That's called
Let's
suicide.
not go there, Mar.
That's called suicide.
Beside
It's only
manufacturing.
11.
It's only what?
It's only 11. Let's not go
Let's
there.
not go there.
Yeah, let's not go there. Let's not do our
You know, there are other
Let's
things
not ask
like
those questions.
Yeah.
I got concerned. Arsenic, isn't that a poison?
Isn't arsenic poisonous? Let's kill people.
And isn't it like little by little?
What?
Little by little.
The evidence is out if you should take just a pure vitamin D.
Okay, so for 10B, propose a method by which the pyruvate molecule moves across the mitochondria.
What?
Small problem.
A method. For number 10.
It is dried.
There is
It is
some
dried
evidence that
in through the
receiving
membrane.
a certain amount of vitamin D through supplement can help. However, let's say you need 2,000 units. To be able to supplement that in your body, you
Oh,
have
okay.
to ingest about 8,000 units.
Because we dispense a lot of it at high dosage.
Well, you
What did you
dispense
say?
all doses, because it's not
Facilitated
brain,
Same.
you would understand chemistry.
You go with
right?
that one?
So how do you get it into your body? Well, you need exceptionally high concentrations to be able to go across that brain here, because it's going to be really hard to get across that brain. So you need like 8,000 units to get 2,000 units across the membrane.
And that's why it says so.
During the link reaction, the pyruvic acid molecule is decarboxylated. What molecules removed during this process?
But you take that one tweak. I'm pretty sure.
That sounds like some pseudo crackery bullshit.
I think that was like a
Probably
OH
is.
or
Yeah.
carboxylic acid.
I think it's 5.
Decarboxylated. So that means that carboxylic acid has to be involved somewhere.
Are you in 11?
Yeah, I'm on 11.
During the link
then again,
reaction, the
remember
pyruvic
that,
acid is the
marathon runners have full life expectancy too.
carboxylated.
That is true.
So
I
a
did read that.
carboxyl
Work out
is,
vigorously. So.
or what molecule? Carboxyl?
Yeah, decarboxylated. I think it probably means removing the carboxylic acid.
And you're probably
Would
not
this
just
be
using said,
the carboxyl?
Yeah, that.
but,
CO2H.
causing your cardiovascular system to have problems and yeah.
And you remove that one.
Moderation.
Yeah.
If you want to go, just don't eat it.
12. Coenzyme A carries the reminder of the pyruvate molecule to the site of Krebs cycle. What is the name of the decarboxylated pyruvic acid? Oh, shoot. Decarboxylated pyruvic acid.
Okay, but the salty fries do.
Wouldn't it be COCH3?
Yeah, I think so.
But what's the name? COCH3? Let me see.
We're starting to figure things out, right?
Wouldn't that be a keto?
Ketone.
Ketone?
Yeah.
Would that be a ketone?
It's like every quarter is the same thing. They'll only build the two because of how they.
How many carbons of the pyruvate molecule remain when it's attached to coenzyme A? Two carbons. When coenzyme A binds to the decarboxylated pyruvic acid, what molecule
is produced?
I think it's like acetyl CoA.
Correct. No. Yes, the acetyl coenzyme A comes off. Yes. So the acetyl is the pyruvate. We cleave one of CO2 off the pyruvate to make acetyl.
The connection between coenzyme A and the acetyl group is weak.
How is
This
this
illustrated in model 2.
Coenzyme A and acetyl group. Coenzyme A and acetyl group. Oh wait, is it no? Which one? So which one here is the decarboxylated pyruvic acid? Is that this one?
Yeah, I think so. Yeah.
Is this the decarboxylated pyruvic acid? That one?
Yes.
Yeah.
Is it a ketone or not?
It is a ketone.
Uh-huh.
So did I just say if we go on the keto diet, where does it enter?
Yeah.
At the linkage step. You don't lose glycolysis.
There we go. We got ambushed.
We started with glucose. We go through glycolysis to end up with 2 pyruvates. But pyruvates themselves are actually still too big. So what I have to do is I have to steal the first CO2 from it. And so I'm going to steal that CO2 and create an acetyl. And we're going to use a coenzyme, and that's going to attach it to a 4-carbon chain. You go from 4 carbons to 6 carbons. Okay? And that is then going to enter into the Krebs cycle. The Krebs cycle goes, so that's linkage step. Okay? So we go from pyruvate to we take in the acetyl, pulls it through the membrane into the intermembranous matrix of the mitochondria, which will do the Krebs cycle. Mar asked an interesting question. What is this called? It's a ketone.
It's a ketone.
So, if you are on the keto diet, where do those proteins and fats enter into the metabolic process?
Linkage.
Linkage
At the
step.
linkage step. So if you are only eating protein and fat, you're on the keto diet, or you're on the Atkins diet, it's the same diet. The carnivore diet, sure. You are entering, your metabolic processes are entering at the linkage step. Therefore, it's skipping glycolysis. So one of the problems with that is how much ATP are you going to produce? More or less?
Less.
Less. You're going to produce 2 per whatever glucose you would have had in there.
Per glucose
Less.
molecule.
Plus you'd have had less NADHs in there as well, which would go into electron transport chains. So you're going to get less. So imagine this. You want to lose weight. How close are you to death at any moment? 2 seconds.
2
You have to
seconds.
produce the same amount of ATP. So if you do not have glucose, where are you going to enter at?
Linkage step.
Linkage step. I'm going to burn fat. I'm going to burn muscle. I'm going to burn proteins. This is a very effective way to lose weight. Absolutely. It is a fabulous way to lose weight. Tell me what is wrong with it.
You're anemic.
You're anemic. You're no longer your blood cells, because the blood cells do not have mitochondria. Your blood cells cannot go through glycolysis. Therefore, your blood will die sooner. Blood cells get repopulated.
And
Okay? You're going to become anemic. And if you're anemic, your body's going to fatigue, including things like your heart and your brain, which uses a lot of oxygen and a lot of electron transport chain. So is this a good thing long term?
No.
No. In fact, if anybody knows the history of these diets, they come and go. Okay? They're very cyclical in our nature. They get renamed. So when I was growing up, the keto diet was called the Atkins diet. Does anybody know what happened to Dr. Atkins, the guy who designed the diet?
He died.
He died of his diet. People die from this all the time. So, is this a good thing to do? Well balanced.
Do you have anything to say about the ethics of the weight loss industry? Like,
Oh, do we want to go there? This is the benefit of tenure. I can say shit like this. I am going to produce a lot of doctors over my career. My goal is to produce not shitty doctors. Shitty doctors are one who want to see you all the time. Okay? I want a doctor who's going to treat you, keep you well maintained, have a good standard outcome, good longevity of life, quality of life. That's what I want a doctor to produce. If your doctor says, I want to see you every week for the rest of your life to do an adjustment on you, they are a quack. I don't want to produce those doctors. So if you want to be that kind of doctor, I don't want to be attached to you, right? So the weight loss industry is full of quacks. I can get you to lose weight very quickly. It's real easy. You stop eating.
True.
Problem.
doesn't work.
Doesn't work. Because I have you blow into a balloon. If you stop eating, your metabolism is going to slow down. But your desire, your all those things are going to actually increase. So now when you start ingesting that material, your body's going to become hoarding because we're going to enter another metabolic pathway because it's thinking you're starving. And so you're actually going to balloon in weight, not lose weight. So the majority of the population actually needs to increase their calorie intake. They need to increase the quality of food. They need to enter these pathways in lots of different ways to actually lose weight and be sustainable. The other thing is, it is perfectly okay to be fat and healthy. It is also
That's me.
very often skinny and not healthy. Being skinny does not mean being healthy. In fact, it's usually the exact opposite. The skinniest people are usually very unhealthy. So I want you to be very, well-rounded, have a great life, all of those things. Yeah. And when in doubt, if they're trying to sell you something, it's usually something that
Yeah, I think the weight loss industry is like evil genius.
Yeah.
It just keeps the people coming back.
Our
It doesn't work.
society has inflicted unhealthy ideals of what. I'm going to tell you a quick little story because we had this conversation. There's this really beautiful Victoria's Secret model, and she was on doing this interview, and we were looking, and there was a group of us. We were watching this thing. Fuck, she looks young, and I'm so bad at judging ages. Okay, I have you guys. I all see you at the same age. You're all just babies. Okay, like
Thank you.
even if you're just
Thank you.
okay?
I love you. I want to change
I'm
that.
so bad at ages. I'm like, but she's in like, it's obsessive, like talking about her diet is why it came up. And like controlling every little thing. And you start paying attention to her, she's getting skinnier. I'm like, how old is this poor girl? She was 14 when she entered the Victoria's Secret modeling. So our society has created unreal expectations of what beauty is and what maturity is and what sexualization and all these things. And we're going to cover some of that stuff as we do physiology. And so we're going to start thinking about it. And so this is actually great questions because next quarter is physiology. How to be healthy, how to grow organs, how to do these things and maintain a good standard comes back down to the chemistry. So we need to have a good understanding of today so we can understand health and wellness in the future. So great question. Yeah, drives me nuts.
I agree with everything you said.
Yeah.
No keto diet.
No keto diet.
Please.
You know what? If
No keto diets.
meet an actual dietitian, meet a real wellness experts, and they're going to help you. They're going to help you get along. And your ideal of what beauty is might not be achievable. And that's perfectly fine. None of you are going to be Tom Brady either. Okay? Not a single person in here is going to be a professional athlete. And why can I say that? Because you aren't already. I'm sorry.
Damn.
Damn it.
I could be,
But
but not ready.
what can
Have
you
that
be?
hope.
Decent person who's healthy, who lives not only a long time, but a good quality of life. Those are independent of each other, by the way. Okay? So I want you to have a quality of life and longevity.
Ooh, yay.
Right?
Can I recommend a diet?
No.
Is vitamin T?
I don't want to know. Let's move on.
It's a taco diet.
The talk. Okay. Yes.
A taco diet.
I'm on the seafood diet. I see food and I eat it. Right?
I'm on the land animal diet. I eat beef and chicken mostly. The only seafood I ever touch is tuna. I don't eat shrimp or salmon.
You don't eat salmon?
I don't eat salmon. And every time I eat it, I'm like, there are times where I do eat salmon, and I'm like, ugh. what the hell did I just eat?
Oh, really?
Yeah, and I just get all these weird smells in my mouth, and it just makes me.
It's just too strong for you.
Yeah,
It's too strong the flavor, right?
yeah, it's a strong flavor. It's a,
But you don't get any
it's like a
allergy
strong-
reactions, do you?
No, not an allergic reaction, but I just get this bad.
So it says, where in the cell does the Krebs cycle take place? In the middle? Do we say in the middle or what? Do we say?
Oh my god. Garlic bread and no.
Would you say in the middle?
Really actually good croissants. I swear it's so good. I feel like I started like getting to real Oh, the hot spring. And there's no difference. It's not so crazy. So like, we're sitting there drinking. Why do you think I'm buying this food?
What molecule is introduced in the Krebs cycle from the link reaction? Oh, wait, did you put in the middle or not? Number 10, number 16.
It looks like it's taking place in the inner matrix.
Oh, that's the inner matrix.
Yeah.
You know what? I think I skipped the last page, right? You were doing
Yeah,
the
you
last
did.
page.
Yeah,
I'm
you
so
did.
sorry. Yeah. Okay.
I answered A, B, and C.
A, B, and C.
The connection between coenzyme A and the acetyl group is weak. How is this illustrated in Model 2?
Is that C or? Oh no.
That's
That's D. Okay.
Oh, that's D, okay.
Okay. How is this illustrated in Model 2? I don't know.
What did you answer for 12D? Connection between coenzyme A and acetyl group is weak. How is this illustrated in Model 2?
Ketone. Which is probably the same, right?
Number 12.
I know it's like the same.
12D.
Yeah, 12D.
One of my best friends we go to Skyway.
Let's go to the next one. Has there any ATP been produced? Used or produced?
Has any ATP been used or produced annually?
Did you say yes? No, that's pyruviate. So this is after. Is that D?
Yeah.
Oh, we'll ask that one. For 13, has any ATP used or produced during the link reaction?
I haven't answered that one. I haven't gotten to it yet.
I think no, because well, there's two APs produced there after.
There's 2 ATPs produced in after, so the answer is no.
Not during, right?
No, not used or produced during.
Not during. Have any other high potential energy molecules been produced during the link reaction?
NADH. No, NADH. FADH2.
Huh? NADH and FADH? From where to where would be the link reaction? That's the question. Only FADH2?
Yeah, I think so.
Is that what you're saying? On number 14? He's got FADH2. Why not? How many acid TOA carbon dioxide and hydrogen are produced in the link reaction for each decaysal?
How many acetyl COAs, carbon dioxide, and NADH? For that question, I said 2 acetyl COA, whoa. Looks like that's 6 CO2s total.
Huh?
It looks like from that reaction, there are 6 total CO2s.
And acetyls? Did you put 2?
And acetyls, I put 2.
Acetyls. God. No. And 1 NADH? No, 6.
I said 8 NADH total.
Oh, 2 and 6?
Yeah.
Okay, so 16. You said inner matrix.
What molecule is introduced into the Krebs cycle from the link reaction? FADH2. Is oxygen needed as a reactant in the Krebs cycle?
Oh my God. What did you say?
I was gonna say no, because it looks like there's no standalone oxygen. The only oxygen is in the compounds. Oxyl oxygen. 1, 2, 3, 4.
You know, statistically, what's the worst thing you can do?
1, 2, 4, 5.
Well, what up? Oh. So you say no? Did you say no? So 18? For 18, what did you have? Yes or no? Because I see this as a reactant. I see the CO2, but over here.
Yeah, I said no because I said
Comes from like
Okay.
think is as without an acceptor.
As a what? An acceptor.
Acceptor of electrons. In ETC, I think that's the only time we actually put oxygen
In ATC?
ETC.
ETC.
The electron transport chain at the end, oxygen accepts the electron and then it's turned into the carbon. With cellular respiration. That's the only time we should put oxygen into the cycle.
Okay. Thank you. Compare the oxaloacetate molecule with the citrate molecule. How many carbon atoms are in oxaloacetate? Carbons in oxaloacetate. 1, 2, 3, 4. Six. Do you have 4 and 6?
Yeah.
Making sure I counted my carbons.
Yeah, I counted your carbons.
And where did they come from? From the
I said from the acetyl
acetyl,
CoA.
right? From acetyl. Acetyl CoA. Okay. 20. Compare the citrate molecule with the
Alpha ketoglutarate.
how many carbon atoms? A what?
Alpha ketoglutarate.
Glutarate.
How many?
Carbon?
1, 2, 3, 4, 5 carbons.
Where did the missing carbon atom go?
CO2.
Compare the molecule
Compare
with the
the alpha
succinate.
ketoglutarate molecule to the succinate molecule. Succinate. Succinate 4.
1, 2, 3, 4.
Where did the missing carbon atom go? CO2 again.
Throughout the Krebs cycle, energy is transferred to the
And it's like, okay. I cannot be used all of a sudden. And then my little trackpad gets like stuck. Yeah, I'll take the
Like when you see these, what are the high potential molecules?
I said citrate is the highest. Oh, I think it's talking about like the NADH, ATP, and FADH, is like the high potential.
NADH, NAD plus? No, just any.
I think NADH. I'm not sure what counts. I was just thinking NADH, FADH2, and ATP are the high potential.
NADH
That
and
would be alpha
FADH2.
ketoglutarate.
High potential.
And then systemic rocks or whatever. That was my guess because I saw a lot of, it's like one like a lot of reasons. Yeah. I wanted to say a lot of other. I don't know.
Throughout the Krebs cycle, energy is transferred. How many produced though?
Alright, we're just going to go through this real fast because we're going to cover this a lot in lab and we'll finish this up. So, this is called the Krebs cycle. The Krebs cycle is going to begin with this acetyl coming in. The coenzyme here is just going to come off, okay? And we're going to hydrolyze it. We're going to add water to the system as well. So we're going to go from 4 carbons, 1, 2, 3, 4, to 1, 2, 3, 4, 5, 6. He doesn't put those 2 carbons. We're just going to count our carbons, okay? So 4 to 6, and we're going to do a rearrangement with water, and then we're going to remove a
CO2.
CO2. Where does that CO2 go? Out to the environment. Every time we get rid of a CO2, we're going to make an NADH. Okay? We're going to get 5 carbons now. We're going to remove a
CO2 and make
and
an
we get
NADH.
NADH. Okay? We're going to do a rearrangement. We'll talk about this in lab here. Essentially, I'm just going to, because I have a high energy bond, I'm going to steal an ATP molecule. It's just going to be, it's called substrate level phosphorylation again. I'm going to steal it because I'm doing a rearrangement. And I now have 4 carbons. Because I have these 4 carbons, I can do some fun rearrangements because of the polarities of them and get another FADH, another NADH, reset it, add the acetylcholine, come back, do the Krebs cycle again. So for every pyruvate, the Krebs cycle goes 1 time. Every glucose, it goes 2 times. Okay? So substrate level phosphorylation is literally, I just get ATP. I did that in glycolysis and I do it in Krebs. So per glucose, what is my net ATP? 4. Because I get 2 in glycolysis and 2 more in Krebs, so that's 4. That's cool. Then I'm going to produce all these NADs and FADHs. Where do they go? To the electron transport chain, which I'm going to use to build my proton gradient, which then I'm going to use to make
Water.
ATP.
ATP.
Okay, through slamming them together. We'll cover all the rest of this. So when you eat a glucose molecule, you have 6 carbons. We lose 2 at the linkage step. Okay, then we lose 4 more because this happens twice in the Krebs. You start with 6 carbons, you leave with 6 carbons. So let's say you want to lose weight. What do you need to do? No. You work out. Because if you work out, you still need the same amount of ATP, but I'm using that ATP to do work, so I need to maintain at a higher. So I need to burn more glucose. Now let's say you're running low on glucose in your body. Okay, you didn't eat, or you ate a well-balanced breakfast, but now you're running low on calories. Okay, we're running low on energy. Well, if you want to lose weight, it's going to steal from your fat reserves. But what you don't want to do is steal too much, too quickly, or inflate your, heart, all those fun things. Right? Yeah, sorry.
Question 22.
Don't ask me a real question.
Energy molecules, is that the NADH, I think, or is that like talking about the citrate that alpha group?
Okay, throughout the Krebs cycle, energy is transferred. Yes, this is the NADH and FADHs.
Okay.
Amos, Benson. Yeah, Amos, Benson, Andre.
Hams.
What?
I certainly did 4 on mine, I think.
You did good?
I said I did. I think I did 4.
I did bad. I thought I had that right.
I feel I did better on this one.
How much did you do in the last one?
Haley, Allison, Vivian.
But I forgot to answer one of the questions.
Oh.
Because you ran out of time?
No, I turned it in too early.
Oh.
I was going back and forth.
Elijah,
And you thought you had it.
Anna. I just handed you the wrong one. My bad. No. I didn't call you.
I'd love to have mine back.
Ashby. Carter.
Carter.
Amber.
109.5.
Not bad.
Not bad.
Hi.
I thought I got like 90 or 80 or something.
119.
What did you get?
Oh my god, this is horrible. Yeah.
What did you get?
No, 119, but
119.
Juan.
But I counted 3 times 3 equals 6. I didn't do the math, like 3 times 2 equals 6. I would have known.
Yeah.
It was 3.
What?
Katie?
I think I got the meiosis one right.
I cannot believe it.
Yeah, I can't believe it either.
Because I know that one. I just, my math was off.
So the right answer was no. Okay. Well, release factor. Okay, so the answer was no. I circled yes and I got it wrong. Okay.
Different person. Different person made it easier.
I got 14 out of 15 on question 8. 6
Scary.
16.
Fun. Like I just saw you.
Oh, I got question 9 all the way right. Oh, so it's the.
Daphne?
Oh, it was the last one that it was
the
like.
last one that I got.
Diana?
The last one that I had.
I
an
think
issue
everybody
with.
got the last
And then-
one pretty bad.
And the. Check it.
Jane.
Mate. Oh, but it's good.
Grace. Amy. Monica.
Wait, is this correct or not?
Jasmine M. Jasmine M, right? Isabel.
When you put a 1, what does that mean?
Ruby.
The 1.
I think that's just saying you
W.
have to commit.
That's you.
Who needs cocaine, you guys?
Isaac. Lena.
I know! Actually, is this right?
CC, Natasha.
Remember they were all like
Alyssa's not here.
Yeah, I did the same one. I did that
What did you get? I don't know. Oh, you're coming at me.
Okay.
I got these right. I wasn't sure.
Who's Miss Sudaika? What?
When you put a 1, what does it mean?
Wait, Vincent, what did you
You got partial credit.
Partial.
really good? Okay.
How'd you
Okay,
do?
never mind. Thank you.
That's good.
Good enough.
I got 109.5. Not my best exam, but I'll live with it.
Could have done worse.
I could have actually done
I
better.
got a 1.
Oh,
No.
I mean, I got
I could have
119.
actually done better. I could have gotten